Okay, welcome everybody. This is the histology module for online A&P 1, and we're going to deal with the next level of organization above that of the cell, that is tissues, which are groups of cells acting as a group or as a team to perform a function that a single cell in the human body alone could not accomplish. So, you're going to define histology and identify the characteristics and classify the functional and structural types of epithelial, connective, muscle, and nervous tissue. You'll also identify and label components of each type of connective tissue, and you'll compare and contrast the three types of muscle and outline the features of nervous tissue. Once you're done with this lab, you should be able to define histology and ID the four main types of tissues, ID the structural types and functions of epithelial tissue, including simple, stratified, pseudostratified, squamous, columnar, cuboidal, and transitional. Describe 5 different types of epithelia categorized by function, including ciliated, exchange, protective, secretory, and transporting. Identify characteristics that describe connective tissue. Classify 4 different types of connective tissue and describe their function. Describe muscle tissue structure and function. Describe nervous tissue structure and function. Explain the histology of each type of functional epithelial tissue. ID and label components of each type of connective tissue. Compare and contrast 3 types of muscle, and outline features of nervous tissue. Now, this is one of the first labs you'll be doing that's going to be entirely virtual. Okay, so you're going to access the cloud available via Hands-On Lab. You'll be using the vScope. The website will be posted on the page on Canvas, and what you're going to need are the following online slides from the online slide box: adrenal gland, dense connective tissue, loose connective tissue, pancreas, pituitary gland, pseudostratified ciliated columnar epithelial tissue, reticular connective tissue, simple columnar epithelial tissue from duodenum, simple columnar epithelial tissue from stomach, simple squamous tissue from lung, stratified squamous tissue, stratified non-keratinized squamous epithelial tissue, and thyroid gland. To fully and accurately complete all these exercises, you'll need access to a computer to upload your digital images. Now, normally in the past, with working with the models and the other materials, you've been taking digital camera photos. Here, you're actually going to be saving images online and including those in your lab report. So you'll be able to capture those photos as you look at them. You'll need basic photo editing software such as Microsoft Word or PowerPoint to add labels, leader lines, or text to digital photos. And you'll need material from the lecture side of the course, including material from the text and any appropriate reference resources that aid you with the background you need to know in order to complete this lab properly. So, histology is the microscopic study of the anatomy of tissues. Groups of cells that work together to perform a function are called tissues and are held together by cell-cell junctions. Studying cells and tissues at a microscopic level is important to understanding the functions of organs and organ systems. There are 4 types of tissue in the body: epithelial, connective, muscle, and neural tissue. You can see examples of each here. So you might wonder, well, what am I looking at in each of these slides? Well, in the lower right, you're looking at nervous tissue. You can see neurons, which are the cells that send and receive electrical information here and here, okay? You can see glial cells, which help the neurons with functions they're too busy to carry out on their own. Examples in the central nervous system include things like oligodendrocytes, ependymal cells, astrocytes, and microglia. Over here you can see smooth muscle tissue. This is an example of a tissue that can do one thing that no other tissue can, which is contract. So it can change chemical potential energy into kinetic energy, the energy of movement. Over here you see loose connective tissue. Okay, connective tissue, as the name implies, attaches different tissue types to each other. Okay, connects tissues together. Connective tissue is probably one of the most varied tissue types that we'll look at. Examples include bone, cartilage, connective tissue proper, and blood and lymph. And over here you can see an example of epithelial tissue. Epithelial tissue is designed to generate body coverings. It also secretes and absorbs material, and it also serves as a barrier, both physical and biological, so that there are components that cannot penetrate this layer unless the membrane properties of the epithelial tissue allow that to take place. So we start with epithelial tissue. It protects the body by forming a layer of cells that act as a barrier between internal and external environments. Epithelial tissues cover and protect surfaces that are exposed to the internal or external environment. Examples of epithelial tissue include skin and the lining of the respiratory tract. Epithelial tissue may also line cavities, vessels, and glands. All epithelial cells are connected to one another by the underlying tissues by a matrix called basal lamina, as shown in the figure. It's made up of protein filaments that anchor the epithelial cells to the underlying tissues. Between the epithelial cells, there are either leaky or tight junctions. Leaky junctions allow for the free movement of some substances from one side of the epithelia to the other, such as water or ions. An example of a leaky junction is between the cells of a capillary. Tight junctions create a barrier between epithelial cells to keep out unwanted substances. Thus, in order for substances to pass through the epithelia with tight junctions, they must pass through the cells themselves. An example of a tissue with tight junctions is the lining of the digestive tract, as well as the stratified squamous keratinized epithelia of the skin. Okay? Epithelial tissues are structured based on their function. Some epithelial tissues may have just one layer, which is simple epithelial tissue, and some may have many layers, such as stratified epithelial tissue. The shapes of epithelial cells also vary. Some are flat like a pancake, so they're termed squamous. Others are cube shaped, so they're shaped like a box and termed cuboidal. Others are shaped like a post, and so they're called columnar. Transitional epithelia consists of multiple layers of epithelial cells that can contract or expand, such as in ureters or in the bladder. In addition, epithelial cells may have extensions of the cell membrane to increase surface area, such as villi or microvilli, or they may have cilia, an extension of the cytoskeleton that beats rhythmically in order to move substances. And you can see examples of each of these here. Okay, if we wanted to categorize each of these based on the drawing, we could do that. We can start in the upper left, and we'll go with blue here. And this would be an example of simple squamous. Simple squamous epithelia can be found in places like the lungs and also in the kidneys, where it forms the parietal endothelium of the Bowman's capsule. It's designed for diffusion and absorption, and to a lesser extent secretion. This is simple cuboidal. Frequently, simple cuboidal epithelia will have apical villi to increase surface area. This tissue type is involved in secretion and absorption and is found in regions such as the kidney tubules, where it controls the composition of the filtrate that will eventually become urine. Over here, we can see simple columnar. And you can also see apical villi here. Simple columnar epithelial tissue is involved in secretion and absorption. Places where you can find it include the small intestine, where it's located in the duodenum, the jejunum, and the ileum. Sorry. Down here, stratified squamous. Now by stratified, we mean more than one layer, okay? But notice that not every layer is flat like the layer up here in simple squamous. Some of the deeper layers look almost cuboidal. So you always want to refer to the most superficial layers to get the shape of the stratified tissue, okay? Here's an example of stratified, or this could be transitional here. Okay. Transitional epithelium has the ability to stretch and snap back. You could, in a sense, take these two drawings to represent two different states of transitional epithelium. In its distended state, which would be here, it looks like stratified squamous. In its non-distended state here, it looks like stratified cuboidal. Okay, it has the ability to stretch and snap back. It's found lining the bladder, the ureters, and other components of the urinary system. And then over here, you have pseudostratified columnar epithelium. Notice the cilia on the apical end. Okay, it is a simple tissue, even though it doesn't look like one, because some of the cells are short and the others that are taller lean over the shorter cells and give the appearance of a tissue that is stratified. Places where you would find this would be the upper respiratory, where specialized cells called goblet cells, shown here, secrete mucus, while the cilia propel the mucus away from the lungs and towards the oral cavity. The purpose being to trap particulates and pathogens so that they don't end up down in the lungs. Now, the value of understanding the arrangement of the cells comes from understanding the function of the tissue. There are 5 different types of epithelia categorized by function: ciliated, exchange, protective, secretory, and transporting. Each has a specific function which are supported by their histology, and the table shows you the type of epithelia, the structure, the functional structures, and where they're located. So if we start with ciliated, the basic structure, they're going to be cilia on the apical end. That's the end that faces the lumen of the organ, or the surface, outer surface of the body, as an example. The shape of the cells could be cuboidal or columnar. They generally occur in a single layer, so they're simple. The functional structures include cilia to move substances along a tract or a pipe. And where do we find it? Female reproductive tract, upper respiratory tract, among a few other locations. Exchange epithelia. Shape, simple squamous. Pores present between the cells allow for easy exchange of substances. Where do we find this tissue type? Lungs, and lining the capillaries. Okay. Again, form follows function here. So the reason for a simple tissue type is to allow easy diffusion from one side of the tissue layer to the other. If this tissue were any other shape, or if it were stratified, that transport mechanism would be inhibited. Protective epithelial tissue. Example, stratified squamous. The cells are tightly connected to each other. We find this on the superficial surface of the skin and lining the oral cavity and the throat, as well as the rectum and the anus. Transporting epithelial tissue is either simple cuboidal or simple columnar. The cells are tightly connected to each other. The surface area is magnified or increased by the presence of microvilli. Examples of locations for this tissue type include the intestines, the kidneys, and some exocrine glands. And then finally, secretory epithelial tissue can be either simple or stratified columnar. These structures in secretory tissues are designed to generate some sort of molecule inside the cell and then actively transport it to the surrounding extracellular environment. Okay, so we're going to see protein secreting and steroid secreting cells. Types of transport include things like vesicular transport and other types of active transport, such as symports and antiports. And where do we find these tissue types? In exocrine glands, such as sweat, saliva, or pancreas, and in endocrine glands, such as the thyroid, the gonads, and the adrenals. Ciliated epithelial cells beat rhythmically and move substances along a tract. These cells line the respiratory tract to push unwanted foreign substances that enter the body through inhaled air or superiorly towards the mouth. There they can be swallowed and then disposed of through the digestive tract, or they can be spit out through the mouth. These cells are pseudostratified because they look as if they are stratified when in fact they are not. They are in reality a single layer of epithelial cells as shown in the figure. Ciliated epithelial cells also line the fallopian tubes of the female reproductive tract and help propel eggs on their way to the uterus. Exchange epithelial cells are thin and flat, so they're squamous in shape, and allow substances such as gases like oxygen, CO2, and other gases to be passed easily across the epithelium. In addition, exchange epithelial cells are leaky, allowing small substances like water and small molecules to pass through gaps between the cells. The lungs and the capillaries are composed of exchange epithelia. You can see an example here in the kidney where the simple squamous epithelia located here produces the parietal endothelial layer of the Bowman's capsule, and then you've got in the capillaries of the Bowman's capsule simple squamous epithelia forming part of what we call the transport membrane in the kidney, okay, which include the capillary endothelial cells, the basal lamina, and then specialized cells called podocytes that form the superficial aspect of the visceral endothelium. And this in the kidney forms a selective filtration mechanism that only allows substances below a certain molecular weight to enter the filtrate and keeps in the blood things like proteins, red blood cells, white blood cells, platelets, and so on, as well as large plasma proteins. Protective epithelial cells are stratified with tight junctions to prevent any substances from passing through the epithelium. The skin is an example of a protective epithelia. The skin prevents exchange of substances from external and internal environments. It includes keratin, the same tough structure present in hair and fingernails. Protective epithelia are located on the linings of the mouth, the esophagus, the pharynx, the urethra, and vagina. Protective epithelial cells are exposed to bacteria and other foreign pathogens on a regular basis, so the lifetime of protective cells is short. Cells located just above the basement layer of the protective epithelia undergo rapid mitosis to generate new cells. As these cells are produced, they push the old cells upward towards the surface of the epithelial layer. Keratinized cells eventually die before they reach the surface. The remnants of cells are on the surface protect the cells below it and eventually are sloughed off. So this is a wonderful example of how form follows function. So you can see down here in the deeper layers of the epidermis, the stem cell layers, okay? These divide, and when a stem cell divides, something very interesting happens, which is that, there are cells that are going to remain attached to the basal lamina in the underlying dermis that will remain stem cells, while the other cells will differentiate and move upwards, and as they do, lose contact with the oxygen and nutrients in the connective tissue below it and begin to die. And as they die, they begin to shrink. As they shrink, they take on a spiky appearance, hence this layer called the stratum spinosum. And then as they continue to move further away from the blood supply and the nutrients, they begin to form keratohyalin granules, and the cells begin to lose a lot of their organelles, and this forms a very tight waterproofing layer that inhibits diffusion to the cells above it. And so once the cells pass through the granulosum layer into the lucidium and the corneum, they're officially dead. But the dead cells are important because they provide a physical and biological barrier against pathogens, and as they're shed, the pathogens that are on the superficial surface go off into the environment and they don't get a chance to set up infection. So it's an excellent example here of how form follows function. These cells are designed to eventually grow up and to die. Transporting epithelial cells selectively transport ions and nutrients from the external environment to the internal environment of the body. These cells are found in the kidney, where they are simple cuboidal, and in the intestine, where they're simple columnar. Transporting epithelial cells have a thick cell shape and act as a barrier to the external environment. Tight cell junctions prevent the movement of substances between the cells, while villi are there to increase the surface area of the cell that's closest to the lumen, and this allows more available space for transport and secretion. Extra mitochondria provide additional energy for active transport, and you can see an example of the tissue in this figure. Secretory epithelia include cells that synthesize and secrete substances like mucus or hormones. Secretory cells may be embedded in other epithelial cells, or they may be clustered together in either an exocrine or an endocrine gland. An exocrine gland secretes substances to the outside environment, normally using a type of active transport. Examples include sweat, saliva, or stomach acid. Goblet cells are a type of exocrine cell that secretes mucus. In the figure, you can see examples of the goblet cells embedded within the secretory epithelia. An endocrine gland also secretes substances, but it secretes them into an internal environment. The secreted substances are called hormones. All secretory cells are modified epithelia. Okay, so you can see, again, another example here of a secretory epithelia. Now, again, what is the function of mucus, which is the product of the goblet cells? Well, it's to protect the lining of the tract, to trap pathogens, okay, and to lubricate materials that are going to move through the tract. So very important function for the goblet cells. Now, that said, these cells are also going to die and be replaced. Okay, so they are usually found to be very actively mitotic, and the reason for that is that they undergo quite a few chemical and physical insults. Examples of where you might find these type of secretory cells with the goblet cells and so on, lining the upper respiratory, lining the digestive, okay, and also lining portions of the conjunctiva of the eye. Another category of tissue in the human body is connective tissue. It provides support for internal organs and tissues. Connective tissue is characterized by its extensive extracellular matrix, which consists of water, proteoglycans, and protein fibers. Connective tissue may be described as fixed if it does not move and mobile if it flows around the body. Most connective tissues are fixed. Types include loose connective tissue, dense connective tissue, adipose tissue, blood, and supporting connective tissue. The elastic loose connective tissues lie under the skin and are also found around small glands. Reticular connective tissue, a type of loose connective tissue, consists of reticular fibers that provide scaffolding for other cells from lymph nodes and bone marrow. Dense connective tissues are elastic and strong. Dense connective tissues include tendons, ligaments, and the tissues that cover muscles and nerves. Adipose tissue consists mainly of fat in liquid form, which occupies most of the cell's volume. Most of the adipose tissue in adults is called white fat. Brown fat is an adipose tissue that helps regulate temperature in infants. An example of adipose tissue is shown here. Blood is a mobile connective tissue, and it, along with lymph, has an extensive extracellular matrix. In blood, the extracellular matrix is called plasma, and in lymph, it is simply called lymph. Okay. Finally, supporting connective tissues include cartilage and bone. Cartilage does not have a direct blood supply, but through its extracellular matrix, nutrients diffuse into the cells and allow them to continue to survive and maintain the matrix around them. One thing to notice about connective tissue in general as compared to epithelial tissue is that connective tissue has an extensive extracellular matrix while epithelial tissue does not. So they're almost polar opposites of each other. If we were to look at epithelial tissue, we would say little to no extracellular matrix, many cells. Well, if we talk about connective tissue, we have abundant extracellular matrix and few cells. Muscle and nervous tissue are unique. Both muscle and nervous tissue are excitable tissues, meaning that they are able to create electrical signals known as action potentials that are transmitted through this tissue. Muscle tissue is characterized by its unique structure, including contractile filaments, actin, and myosin. These filaments, under the correct conditions, contract to induce force and ultimately help to move the body. There are three types of muscle tissue: skeletal, cardiac, and smooth. You can see examples of each here. Okay? Skeletal muscle is striated and under voluntary control, meaning that we consciously tell it what to do. It's attached to bone. Smooth muscle is found in the linings of organs and blood vessels and lymphatics and responds to hormones as well as nervous signals and local reflexes. And it gets the name smooth from the fact that it lacks striations. Cardiac muscle is striated and involuntary and only found in the heart. Now, you might wonder, what's the big deal about these striations? Well, the striations represent areas in the muscle where the contractile proteins actin and myosin overlap. Okay, so if we were to diagram the layout of the contractile units in cardiac and skeletal muscle, and we'll get into more of this when we get to the muscle chapter, what you would see are thick filaments. Draw these here in blue, thick blue filaments. Attached to each other by a structure called the M line. And then interdigitating with those, we're going to have thinner filaments, and I'll just draw them as thinner. I would like to. They look like more like this. I'd like to draw them a different color. Can't pull the color up right now. They're attached by a structure called the Z-disc. The thick filaments are made of myosin. The thin filaments are made of actin. Okay, and what happens when muscle contracts is that the actin filaments are pulled towards the center of this structure, which is called the sarcomere, by the myosin filaments using ATP as an energy source. So here would be like the contracted state. Okay, and this is called the sliding filament principle or theory. Okay, so when we see these striations in skeletal and cardiac muscle, the dark vertical bands within the striations are where the overlap occurs, and the light color bands are where there is no overlap. So when muscle contracts, the thickness of the light band shrinks, and the entire muscle cell will shorten as these complexes of contractile protein inside the muscle shorten and exert their force on their attachments to the muscle cell membrane. Okay. Nervous tissue includes neurons, which are nerve cells that transmit information from one part of the body to the other. Neurons stimulate the muscle cells to move and are the main cells of the central processing units of the body, the brain, the brainstem, and the spinal cord. Glial cells are also considered nervous tissue, cells that surround neurons and provide biochemical support for them. And so you can see some examples of some central nervous system tissue here. This is a motor neuron. Okay, these extensions are called dendrites. This is the soma, and this is the axon. And you can see that it's insulated and protected by these extensions that come off the oligodendrocytes. And the purpose of this insulation is to both speed up the electrical signal and also to protect the axon. Okay. The astrocytes are there to provide nutrients from the blood to the neuron. The microglia defend against pathogens and clean up dead and dying cells. And we also have in the central nervous system ependymal cells, which create a liquid called cerebrospinal fluid that bathes the neurons and allows them to function, sending and receiving electrical information. And I believe that covers just about everybody in the CNS. In the peripheral nervous system, the glial cells include satellite cells, which surround and protect nerve cell bodies, and the Schwann cells, which have a function similar to what the oligodendrocytes do for the axons in the peripheral nerves, which is to insulate the axons, protect them, and to speed up the electrical signal.